everyone, I hope and I believe you all are doing great. This is Dr. Arya, your pathology faculty. Today we will be studying the topic of glomerular diseases. Okay, let's start. So what are glomerular diseases? Glomerular diseases is a tuft of capillaries. You have something called as the Bowman's capsule. You have something called as the Bowman's capsule. So in the Bowman's capsule, you have a tuft of capillaries. That is nothing but your glomerulus. Okay. So they basically serves as a primary site for filtration. They are like the ROs that you have in your home for the water purification, and they are the ROs for your kidney for the purification. Now, let's see the structure. You have the endothelium of glomerular capillaries. That is, first thing, you have the fenestrated capillary endothelium. That is approximately 70 to 100 pores, and it prevents the passage of RBCs, WBCs and platelets. So from this structure, no RBC, no WBC, no platelets will be able to transfer. They are covered by a negatively charged glycocalyx, which repels the negatively charged plasma protein. So you know, like positive, negative, attract, negative, repel. That is why you will always see, whenever you like a person, there will be something opposite in that personality that you are not having, and you both just get repelled to each other, because positive negative attracts and negative repels, and positive also repels, right? This is the basic fundamental of life as well. Then you have the glomerular basement membrane. It is trilaminar. It has the layers. First one layer is your lamina rara entera. Entera is the inner one. Then you have the lamina densa. Densa is the middle one, and then you have the lamina externa. So interna is adjacent to the endothelium. Densa is adjacent to the collagen and lamina, and externa is adjacent to the podocytes. Podocytes are for the movement of the cells. Okay, they act as a charge and size barriers. Diseases. What are the diseases you have? First one is your good pasture syndrome, the GPS syndrome. Their autoantibodies are produced basically against a A3 chain of type 4 collagen. Then you have second something called as the Alport syndrome, and don't worry, we will be reading that in details also. Okay, in Alport syndrome there is mutation in the type 4 collagen. Then you have the diabetic nephropathy, that you all know. There will be glomerular basement membrane thickening, and there will be a mesangial expansion, right? Next you have the podocyte. They are the specialized cells covering the outer surface of the GBM. Like this is your GBM. On the outer side, you will be having some podocytes. You will be having some podocytes. Okay, because if the podocytes are not present, you will be having the disease. If the podocytes are not present, you will be having the disease called as the minimal change disease or the focal segmental glomerular disease. These are basically the two diseases which you will be having if you are not having the presence of podocytes. So podocytes are that important. What is supporting structures? You have the mesangial cells that are present between the capillary loop. They basically provides the support. And secrete the cytokines. Secretes the cytokines which are responsible for the growth and matrix. What is the pathology? You have something called as the IgA nephropathy, which is due to the mesangial IgA deposition. Juxtaglomerular apparatus, the very famous, and I guess we all have read that in our 12th standard as well. So juxtaglomerular cells are the modified smooth muscle cells of afferent arterioles, and they secrete renin. Then you have macula densa. Macula densa is the distal tubule cells sensing the NaCl delivery, the sodium chloride delivery. You have the sodium ATPase pump, right? You remember the mnemonic of Nokia that we have read, right? So the function is the regulation of GFR and the BP via the RAS system. RAS system you will be learning very beautifully in your physiology lectures, okay? What is the physiology of filtration? So you have something called as the forces are involved, called as the Starling forces, right? So here in the Starling forces, the glomerular capillaries hydrostatic pressure is approximately 60 millimeters of mercury. Okay, and the Bowman space hydrostatic pressure is around 15 millimeters of mercury. Plasma oncotic pressure is around 25 millimeters of mercury. So it has to be in that range to maintain the hydrostatic pressure. What is the histology? You have the light microscopy. Normally, you will be able to see the capillary loops, mesangium and the podocyte nuclei. This is the normal. I will show you the figures also. In the pathology, you will be able to see the hypercellularity. There will be increased number of cells that will be there. There will be basement membrane, thickening and sclerosis and crescent shape like structure that will be there. You have the linear deposits in the 𝗲𝗵𝗵 syndrome. We will be covering in details. Okay. Then you have the granular in the immune complex diseases. In the electron microscope, it is mainly essential for the diagnosis of your minimal change disease because there will be podocyte present, and it is also responsible for the diagnosis of MBBS, that you will be able to appreciate a appearance called as a tram track appearance. Okay. In the alport, you will be able to appreciate a basket weave-in appearance that I have told you, right? So we will learn about all the syndromes in details in the next video, because in next video we will be studying about the nephrotic syndrome, the very famous nephrotic syndrome. So if you are having any sort of doubt, please feel free to ask me. Please feel free to reach me. Thank you so much.